child. I'm in my own kitchen today and I'm boiling up some primordial soup. And there is a primordial soup machine. We're doing a recipe for the chemical building blocks of life. Today.
Hey guys, Joe here. Some people would argue the most important year in the history of soup was 1962, when Andy Warhol released his super soupy pop art. But I think soup's best year came a decade earlier in 1952, when a scientist named Stanley Miller first cooked up primordial soup. Miller's experiment took some simple chemicals like those found on early Earth, bubbled them up through a tube, zapped them with electricity, and after a few days floating in this soup, he found amino acids, the building blocks of proteins and one of the essential ingredients for life. This idea that life's origins could be found in a puddle of chemicals is an old one. In the 1920s, two different scientists theorized about life coming from what they called a prebiotic soup. And this soupy speculation even goes back, unsurprisingly, to Charles Darwin, who in 1871 wondered if life may have formed from chemicals in some warm little pond. What made Miller's experiment so special was it gave us proof. Regular non-life stuff could become cool life stuff super easily. But every living thing we see today, even the most basic bacteria, is so complex, built of such intricate machinery, it's impossible to imagine they just popped into existence out of some soup. That's because they didn't. We're gonna go on a journey in search of the origin of life, and along the way there will be a few forks in the road, maybe a couple speed bumps, and we're gonna need help from a couple friends. We'll come to see that Miller's primordial soup isn't exactly how this story.
Okay, now wait a minute. Didn't we debunk this guy last week? What's he doing still on the internet? We totally should have schooled him last week. All this stuff about non-life coming, or life coming from non-life. Super simple. We said that was wrong. So let's get back on track, okay? That's more like it, okay? So tonight, we're going to do another chapter in looking at the design that we see in nature and showing how that points to God. So tonight we're going to do just that. We're going to continue our series of 6 sessions. This is number 2 of 6. You should all have a handout. If you didn't get a handout, let me know. If you didn't get an email this week, let us know, because you should have all gotten an email with a link to last week's video and the notes, the slides, all that stuff. So if you're not getting those, I don't have your email right, check the sheet over there and we'll get that fixed. So tonight what we're going to do is this. We're going to look first of all at how materialists, that's hard to say, explain the world, okay? And we'll find out what's wrong with that. We're going to start with validating a story. How do we validate a story? How do we check and make sure it's true? And then we're going to go to a more workable idea, which is aligned with God's plan. And we're going to go, first of all, we're going to look at an analogy about inside the room or outside the room. It'll make more sense in a minute. And then how do you build life? What does it take to build life? And then we'll do arguments from a mousetrap, and we'll introduce the idea of intelligent design. And then we'll do one more of those long story short videos that helps explain, kind of wrap up some of the stuff we're talking about tonight. But I want to start kind of where we started last week, and that's with my dad. You may remember, you met my dad last week, so I'm sure you're all familiar with him. And he had left some notebooks that were the diary of his World War II exploits. Well, all my adult life, I've been curious about going back and seeing where he was. And we had a conference in Cambridge, England this year, and the place where he was stationed was just north of there. So one of the things we found in his notebook, and he had talked about this all his life, and we were able to validate in the notebook where he was. And it says that he was at Cottesmore, and he stopped on the way at Bottesford and Langer. So there's 3 different places we can check on a map and find out, are those logical? Were there actual air bases there? So we were about 30 minutes from Cottesmore, and we rented a car and went there. And what we found was this cute little village. It's very quaint. You'll notice on the corner of the upper roof, you've got foxes chasing a rabbit, all on a thatched roof. Now, this is like quaint to the max, right? So the village really exists. It's really quaint. It's really cool. By the way, if you can't see, you may want to move over a little bit. I may be in your way, and I apologize. So anyway, we found the village, and we found, we knew that the air base was still active. It was a Royal Air Force base, and they wouldn't let us on the base. So we went there and asked permission. They said, no, you can't come on. And so we filled out a bunch of paperwork. They probably know my, firstborn male children's birthplace. Anyway, but 2 days later, they called the email and said, we got your approval, so you can come on base with an escort. Well, before we went, I had looked online and found this map on Wikipedia of Cottesmore Royal Air Force Base. And this is from 1942. Down here you can see the date on the photo. And so I knew that he was there in 1944, so I knew that we could compare this to today. So we took that map and I compared it to a satellite image of what it looks like today, and we found that a whole lot of the buildings that were there in 1942 are still there. And so we kind of wanted to just go see what that looked like. You'll notice in the original, the black and white photo, there's no runway. So it was a grass big circle that they did all their takeoffs and landings. Until sometime later, I'm not sure exactly when they built that runway. But we found these buildings that we knew were still there, and so we kind of targeted these as buildings that we wanted to go check out. And so we went to the base and we were able to go to the buildings again, but he would have walked around when he was there in 1944 and 1945. So we were able to validate his story because we had evidence. We can do the same thing with other kinds of evidence, and that's what we're going to do tonight. We can investigate the past if we have some kind of data point and there's a way to check it. And so I want to introduce you to a guy named J. Warner Wallace. Any of you heard of him? He's a cold case homicide detective. He's been on Dateline more than any other homicide detective ever, because his specialty is cold case, like it's been investigated and dropped long time ago, but he looks for other clues that other people missed. And so one of his books is called God's Crime Scene, and I'm drawing on that tonight. And so if J. Warner Wallace is investigating a murder, and this is not a cold case murder, this is one if he comes up on a murder scene, he finds a victim, okay? And the first thing he notices is that there is a dead guy, okay? And he's lying face down. So when you come on a scene like this, there are 4 possible explanations for what we see. The first one is natural death, accidental death, and then 3 suicide or 4 homicide. Those only, if you think about it, it sounds overly simple, but there aren't really any other possibilities. So the idea is, which ones can we eliminate, right? So right now, all we've got is this. Let's take a look at our evidence. So the first thing we see is he's face down. Can we eliminate any of these 4 things from him being face down? Probably not. Okay, so let's look at what else we have. Suppose that in this example, there's a pool of blood. Now can we eliminate any possibilities? Which one? Right. So it's not a natural death, right? It is one of the other 3. We don't know which one yet, okay? As we get more evidence, if there's more pieces that we can investigate, what if there's an exit wound on his back? Okay, now what kind of wound is this? Gunshot. So what else can we eliminate anything else here? Probably, but it could still, he could be cleaning his gun, so we don't know for sure. But it's starting to look a little suspicious, right? So we'll get more clues. The next one is, suppose that there's a gun. Can we eliminate anything yet? Not really. It's looking more suspicious, but not really. Okay, now what if there's a suicide note? Can we eliminate anything?
Maybe.
Maybe? Why maybe?
The killer might have written the whole
Right, it could have been just there as a setup, right? Making it look like a suicide. So we really can't necessarily, it's looking like it might be suicide, but we don't know yet. The exit wound's a little weird. Okay, so what if there's now multiple exit wounds? He's either got a semi-automatic he was cleaning, or it's looking more and more like homicide. So we keep adding clues. And so what if there are bloody footprints and they don't match the guy? Now what do we got? Now we have a homicide. And when you have a homicide, you have a situation where, in Wallace's words, a homicide turns curiosity into urgency. So when we get enough clues we can draw conclusions. And that's what we're going to do. So the question is, when we have a situation like this, can we explain everything that we see from what's inside the room? So if everything can be explained from inside the room, for example, the suicide note, the gun, but no bloody footprints, no fingerprints, that kind of thing, we could explain it from just what's inside the room. But if we have footprints that don't match the victim, or if we have too many shell casings, if we have fingerprints that don't match the victim, bloody footprints, like I said, that don't match, then we know that something outside the room has to be into consideration, right? Well, there's a parallel, and when we look at those things and we eliminate and go outside the room, we can do the same thing with issues like the universe. Okay? If everything that we see in this room, in the universe, in the stars, in the galaxies, can be explained from what's inside the universe, then we don't have to look outside the universe for a supernatural cause, right? So if the universe can be explained by what's inside the universe, physical causes, so can physical causes cause the beginning of time, space, and matter? Anyone? Physical causes can't cause physical forces. So the physical forces have to be caused by something outside the physical forces. So when you look at that kind of an analysis, there's a number of things that we can look at. We can look at the beginning of the universe, fine-tuning of the universe, the origin of life, the appearance of design in biology, the existence of consciousness, existence of free will, transcendent moral truths, existence or presence of evil and injustice. These are all things that we can look at and decide, based on the evidence, can we explain it from inside the universe or do we have to go outside the universe? Now, a bunch of these are beyond the scope of our discussion tonight. I'm sure you'll be relieved at that. So we're going to put a bunch of those off to the side, and we'll ignore those. But what we are going to look at is origin of life and the appearance of design. And that's really the focus of this whole 6 weeks, is origin of life and the design of life itself. That appearance, can it be explained by what's inside the universe, materialistic causes, or do we have to go outside the universe instead? So design counter-arguments. Here's where we're going tonight. We'll start with design counter-arguments, how they are, how they're working and so on, or they don't work. So how materialists explain the world, and then we'll go to the more workable ideas. So first of all, how do they explain the world? Well, materialism or physicalism is the doctrine that nothing exists except matter and its movements and modifications. Okay, that's it. Material, atoms bumping into each other, physical forces in the universe. It's a metaphysical thesis, which means it explains more than just the physical. It's a big picture of the world in the same sense that because it tells about the nature of the world being only material. Okay? So you can look at it like this. So here's an example of a cartoon that helps us understand. Climate change is bad enough. Now gravity? So one of the things that the material world can't really explain yet is gravity. We don't know what it is. It's force at a distance. It confused Newton. It confuses us today. There are ideas about, we can describe its characteristics, how it behaves, but we don't know what it is. What is it that causes us to be drawn down to a body that has enough mass? Okay, so positivism, the idea that knowledge can only be based on what is objectively observed and experienced. Empiricism is the idea of doing a hypothesis and doing a lab experiment and validating. That's empirical results. Okay, and positivists would say that the world is a collection of observable events and facts which can be measured, and that's all. Okay, and so once again, here's an example of something that can't be observed. Have you seen my mind? My wife thinks I've lost it. Okay? Our mind exists. Right now, you're thinking something. You're either really annoyed with me, or you like what's happening, or you wish you were someplace else. But whatever's happening in your mind, it's happening. Nobody else knows what it is, but it's real. It's as real as anything that I can touch, see, taste, or smell. But we can't prove it from the physical laws of the universe. So the positivists, based on the existence of consciousness, cannot be correct. Scientism is the belief that science and the scientific method are the best or only way to render the truth about the world and reality. And so once again, here's the mind idea that refutes that. Well, I've been thinking harder than you have, and my thought experiment disproves your thought experiment. Okay? It's all about, how do you know that? It's all a mental thing that that scientism can't get to. And then methodological naturalism, we've talked about this one before. Scientists like to put big words together just to confuse people like us that are just trying to live life. So, but all this really is, methodological naturalism is a philosophical naturalism. Basically, it's just a doctrine that says the only method we're going to use is natural. So we're not going to use any method other than what we can do with experimentation. And the problem is that that's limited to physical entities, and we know that there's more than physical entities. And we'll get into that more in future courses. But if you are looking for something in the wrong place, and it's not physical, in this example, this guy's looking for something in the wrong place. He's never going to find that bird because he's looking in the wrong place. Like looking for your phone when it's in the garage and you're in the house. Have any of you ever done that before? I did it this week, okay? So if you're looking in the wrong place, you'll never find it. And the methodological naturalists, by method, limit themselves to naturalism, and they then are not necessarily looking where the answers may be if they're not a natural explanation. So they eliminate those. So let's look at these examples of these atheist scientists who are materialists. So here's Carl Sagan had his famous quote from the Cosmos. The cosmos is all that is or was or ever will be. Remember, any of you remember that old Cosmos series? How did he know? It was a philosophical statement. It wasn't a scientific statement. Here's Richard Dawkins. The universe that we observe has precisely the properties we should expect if there is at bottom no design, no purpose, no evil, no good, nothing but blind, pitiless indifference. Okay. Neil deGrasse Tyson, the guru of our age of science. I look up at the night sky and I know that yes, we are part of this universe and we are in this universe, but perhaps more important than both of these facts is that the universe is in us. Now, doesn't that sound really whimsical and wise and clueless, right? I don't know what he means by that. And then here's my favorite, Bill Nye. You can't present good evidence that says evolution is not a fact. That's kind of a double negative, but at any rate, he gives us his qualifications too. He says, as you may know, I'm a mechanical engineer. And by the way, I used to work in comedy clubs. But that gig didn't work out so well, so now my new gig is confusing kids about science, right? So these guys are what we would call practicing scientism, with the idea that the only truth we can be sure of comes from scientific investigation. And the problem is that when atheist scientists do this, they become philosophers, and they're not very good philosophers. So there has to be more than they can explain just based on the cartoons we showed. That's enough proof, right? So there's more to life than just the physical. All right, so most of us were taught, if you went to public school, that evolution is the way everything happened, okay? Do you remember this guy? He taught us a little bit last week, and he said, if you hear these phrases like, must have happened this way, surely this was the case, that was two of Darwin's favorite phrases. So we're going to have a short video here that explains the origin of life, and I want you to listen for those phrases, okay? There will be a test. All right, here we go.
4 billion years ago, a ball of rock and dust spins in the frozen vacuum of space. This isn't Mars or Venus. This hell is Earth. This seething, lifeless mass of molten lava will become home to nearly 9 million living species. But generating life from this will take a chain of events that defies the laws of probability. There are many theories. Nobody knows exactly how or where life began. But it couldn't happen without water. Experts believe asteroids or comets delivered it here. The water is churning with chemicals and organic compounds. lightning strikes the chemical soup at the right place and the right time. Billions of volts of electricity trigger a chain of improbable coincidences. The chemical's atoms join up in a precise sequence, creating a bundle of genetic material. These fragile genes don't stand a chance in this extreme environment. But luck strikes again. A blob of oily material engulfs a single chain to create the first ever cell. Now, the genes send out messages, chemical instructions. And 3.5 billion years ago, they do something extraordinary. They copy themselves and the cell to create a perfect clone. This is the very first living thing. Every human, every animal, every bug, every plant can trace its origins to this single cell. The genes tell each cell to reproduce, guaranteeing their survival as they pass from one generation to the next. For 2 billion years, the only living things are simple single cells, but a random accident changes everything. 2 cells merge. Their genes combine. The merged cell clones itself. Its offspring contains genes from not 1, but 2 cells, 2 parents. We call this accident sex. Sex introduces variation. Occasionally, things go wrong. As the cells reproduce, genes get deleted, duplicated. These cells are mutants. Mutations pile up, differences increase, until the cells become so different, they're separate species. The tree of life branches out into billions of species, but only one will lead to us.
Okay, so how many of those wishy-washy, must-have, should-have statements did you hear?
Too many.
Can you think of any examples?
Improbable possibility.
Improbable. How lucky is this? A fortunate event.
Experts believe.
Experts believe. Yeah.
Highest probability.
High probability. And yet, they're really confident, right? So the most confidence you're going to find is in these kind of animated videos or in undergraduate textbooks. And so our kids and we were taught this was a certainty, but it's really not. Okay, so this is like they had a prompt to make this video. Given CGI derive the origin of life. Computer graphics, right? So anyway, I'm not buying it. Let's look at a different way to look at this. This is, anyone? A mousetrap. Okay. Now the idea of a mousetrap with the idea of origins is this. If you have a mousetrap that works, all the parts are functional, they're assembled properly, the trap is set, there's food there's cheese over here, there's baited. But if you had a mousetrap where the base was the size of this right here, it wouldn't be stable. If you had a spring that was too small or too big, if you had any of these pieces not properly sized, not properly assembled, not properly set, how many mice would you catch? You wouldn't catch less mice, you'd catch no mice. And so when we have the idea of incremental small changes leading to big changes, that's the problem, is that small changes don't improve anything until there's a big change. And so the small changes are not selected for. So the very foundation of natural selection, small changes leading to big changes, is false. The guy that came up with this illustration is a guy named Michael Behe. He's a brilliant guy. He's going to explain this in a little bit more detail for a couple minutes. So here's Michael Behe.
Now, it turns out if any of these parts are missing, the trap doesn't work. So not only is it complex, it's what I called irreducibly complex. You can't take a part away and still have it work. If you took away this hammer, the mice don't get caught. Take away the spring or the holding bar, any of the pieces, it doesn't work. Now, the problem for Darwin's theory is that, number one, the molecular foundation of life is run by machines. The cell is run by actual machines made out of molecules. People find that fantastic, but hey, that's the way it is. There are little machines that act like outboard motors that can propel cells along, and other machines that carry cargo from one part of a cell to another. And those machines, just like pretty much any machine, including the mousetrap, have a number of different parts performing different roles, and they're all needed for the machine to work. Darwin's theory of evolution requires that natural selection favor an organism that has a very small change that helps the organism do something better. So if we're taking that view of Darwinism, we can ask, how could something like this, something like a mousetrap, be put together one tiny step at a time? And it turns out it's surprisingly difficult. You know, if you just had the wooden platform, just the bottom, that doesn't catch mice. If you put on, say, this holding bar, you might say, well, maybe if a mouse is running along, it would trip on the platform and impale itself on the holding bar. But that's kind of just silly. So this can't be made gradually. So that's a big problem for Darwin's theory, because pretty much the foundation of life, these things are all over the place. And furthermore, you can ask yourself, well, how do we recognize intelligence? And it turns out that the way that we recognize intelligence is by what I call a purposeful arrangement of parts. And that's when different parts are put up in relationship to each other, where you can see that they have a purpose. That is, the arrangement has a purpose. An easy example are letters in a word. You know, people put the letters in to form a word, words to make a sentence, and so on. And not only is this arrangement, this can't be put together gradually, but we immediately see that the arrangement of these parts has a purpose. And so we immediately grasp the intelligence that was needed to produce something like this. And again, I can't emphasize enough that Darwin didn't know anything about the foundation of life. He and his contemporaries thought that the cell was a little piece of jello, protoplasm, they'd call it. And it was mysterious, you know, did cool stuff, but they didn't know how, so they pretty much ignored it. But modern science has shown that the cell is a lot like a ultra-sophisticated nanoscale factory, far, far beyond anything that humans could produce. And again, they're just chock full of machinery that is much more sophisticated than this simple little mousetrap. So not only have I become skeptical of Darwin, but you can readily see the design in life. So I've argued that many of these machines were purposefully designed.
So the explanation, I think, is fairly straightforward. I think you can get that. So he talked about molecular machines. Molecular machines are things inside the cell that do complex tasks. Here's a short description of some of those.
Look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have two strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little,
so each of those little structures is a protein. And those proteins do mechanical functions. Each organism has its own set of proteins. We're going to get into this a little bit later, but real briefly, a human, the count keeps changing because they keep discovering more, but a mammal has about 50,000 proteins. Every mammal, whether it's a mouse, a human, a beaver, a round numbers, 50,000 proteins. Of those, 5 to 10% are unique to that organism. They have not been found anywhere else in any other organism, which means that each of these proteins, 5 to 10,000 of them, would have had to have been formed accidentally for that organism to exist. And that's just the beginning of the complexity. This is the bacterial flagellum, which is what Michael Behe became famous for describing the complexity of this one little machine. And I'm going to let Stephen Meyer talk to us, I think, about this, the complexity of this structure of every bacteria in our body.
In Darwin's Black Box in 1996, Behe spotlighted and made famous a number of really interesting discoveries that had been occurring in biochemistry and cell biology over the last 2 or 3 decades. And what biologists, molecular biologists, cell biologists, microbiologists have been discovering is that at the level of individual cells, there are little tiny examples of nanotechnology, little tiny machines at work. The flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail, protein tail, that functions like a propeller, and it moves the bacterium through liquid, enabling the bacterium to essentially track down its food supply. And this little machine includes a rotor, a stator, a drive shaft, a U-joint, bushings, bearings, and a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Bacterial flagellum is a true nanomachine, about 40 nanometers in size. It's amazing. I mean, E. coli, Salmonella, which are kind of our model systems for the bacterial flagellum, can propel a cell about 20 lengths per second through a very viscous medium, like water to these organisms. And you extrapolate that to human scale. 20 body lengths per second, 6-foot person, times 20, 120 feet per second. Mark Spitz or Phelps would be setting records with this type of propulsion.
It's hardwired into a signal transduction circuit that allows the bacterium to sense changes in the sugar gradient in the surrounding liquid.
This signal transduction system is actually a short-term memory system where the cell is, if it's going in the direction of an attractant, a nutrient that it can use to metabolize, it follows that chemical gradient. If it's a repellent, it will sense that and move in the opposite direction. So it's more than just this engine.
It's an extraordinary piece of nanotechnology. It's high-tech in low life. And so just by spotlighting these extraordinary pieces of nanotechnology inside cells, and the flagellar motor wasn't the only one by any means, Behe, in a sense, opened up a window for people. He opened up the black box of the the inner workings of the cell and said, look, this is much more complex than anything that the early evolutionary biologists had envisioned. Darwin knew nothing of this type of nanotechnology in cells, and at the very least, you've got to come up
So
with...
we can see that the complexity we're talking about is really beyond comprehension. And the more we learn about it, the more problematic that becomes. So what is the claim that Darwin made? Now, you saw in the first video that things just happen and they get surrounded by an oily cover, right? I mean, the complexity just of the cell membrane is another lecture all by itself. But before Darwin, the prevailing idea was that things were designed. And we had Lyell with his long ages. We talked about that with in our first session on history. And the idea that all of a sudden we think the Earth is now millions or billions of years old. That allows time. And we use our imagination to go, well, anything could happen given that much time. Well, no, it really can't. And now the math has been done. So, but design was the prevailing view until Darwin. After Darwin, evolution without external intervention was the prevailing view. So it's really important to understand why that is not possible, so which we've kind of covered already. So this next video is going to be Stephen Meyer again, just describing, I cut out the part where he's describing the idea of change. There are 3 different kinds of evolution. And he takes about 6 minutes describing that. We're not going to watch that. But he's going to sum up the importance of it. So the 3 different kinds of evolution are change over time. Nobody disagrees that there is change over time. There are minor changes that happen. Finch beaks get small, they get big, they get small, they get big, they're still finches, right? So we have these small changes over time. Universal common descent is another of the claims of Darwinism that cannot be proved empirically. And then there's natural selection and the idea of small changes through to this universal common ancestor, and then the idea of natural selection. So here's what Darwin says about the possibility of outside intervention, just so we understand his theory.
So when Darwin summed up what his book was about, he said it was one long argument for descent, that is his theory of universal common descent, with modification, where the modification is produced by the mechanism of natural selection acting on random variations over a long period of time. Now, now we need to understand why this mechanism gets rid of the idea of intelligent design. Richard Dawkins has written about this in his famous book, The Blind Watchmaker, and he has an admirably clear understanding of this. He says on the first page of The Blind Watchmaker that biology is the study of complicated things that give the appearance of having been designed for a purpose. You might be able to guess what the key word is in that quote. It's obviously appearance, because in a Darwinian view, the appearance of design is essentially an illusion. And it's an illusion because that mechanism of natural selection acting on random variations that we were talking about is understood to be a purely unguided, undirected mechanism that can produce the appearance of design without being guided or directed in any way. Therefore, things look as though they were designed, but they weren't really designed because natural selection has the power to mimic a designing intelligence. Now, science popularizers like Richard Dawkins and Bill Nye, who describe Darwinism this way, aren't misrepresenting Darwin. Darwin himself thought that apparent design was something that could be produced without a designer. In fact, by his mechanism of natural selection. So, for example, he said in a letter to a friend after he'd written the book, The Origin of Species, he says, there's no more design in the variability of organic beings and in the action of natural selection than in the course in which the wind blows. Again, design.
So again, Darwin makes absolutely no allowance for anything mechanical. He believed that his theory, or at least he claimed he believed that his theory was able to explain all of the variation we see in life without appealing to an outside designer. I want to introduce you to another of my favorite scientists. This is a guy named James Tour. He is one of the most gracious and humble men I've ever met, other than Steve Beyer, who was just up on the screen. Both just incredible believers who are really trying to serve the cause of helping people understand the difficulty of design happening by accident. James Tour is at Rice University. He's a synthetic organic chemist. You have to practice saying that. He synthesizes organic chemistry. Now, if you go to med school, any doctors in here? They say when you go to med school, or the pre-med stuff, organic chemistry is one of the deal breakers. Well, he makes these out of thin air, basically, not really, but he synthesizes organic chemistry. He has over 600 peer-reviewed papers, over 50 patents. He does cancer research. One of the things that he talks about is rebuilding cells, like nerve cells. They're hoping to rebuild spinal cords for spinal cord injuries. He builds nano cars and races them and wins. He just does all kinds of things. I'm going to give you a link in the email after this to this talk. I've taken a couple minutes out of it just to get one of the most important points across. But it was in 2019 at a Discovery Institute Science and Faith conference. This is an annual conference all of you should go to if you like this stuff. It's a fascinating conference, and that's the first time I've met this, heard of this guy. I got to meet him later when he talked at University of Texas at Dallas. But he's going to talk about the way we oversimplify the cell and how complex it is in reality. So I want you to listen carefully. He's going to go fast, but he is refreshingly candid. Okay. So here's James Tour.
So this is a cell. What is the origin of life? How do you get a cell like this? What is the origin of something like this? This is an amazing machine. A cell is an amazing machine. It's not just a blob of protoplasm. Every day it gets harder to have the origin of life, to come up with a scenario, because the origin of life becomes more and more complex every day. A cell is a factory. It has the lipid bilayer, which is extremely selective to let certain things in and not other things. It has all of this substructure in here, these little areas where energy is made in here. It has these microtubules which can form so you can move matter from point A to point B. If you go to a factory, what you see is you see these overhead hanging machines that are moving materials from point A to point B. In these systems, and the way they do this is they build these racks, but same thing happens in a cell. You get these microtubules to move material from point A to point B, and then as soon as the material is done moving, the microtubule breaks down and then assembles some other place. You say, well, why doesn't it leave it there? Because then the cell would become too rigid. So it just rebuilds it. It's just amazing factory what's happening in a cell. This is what we have to make. If we want to have origin of life, you got to start here. You don't start here. You start with a single cell. Just build a single cell. That's what we have to do in origin of life. Nobody has ever done this. If you've been taught that simple forms of life have been made, that is a lie that you are believing. Somebody told you a lie. That has never been done. Next slide. Molecules don't care about life. Nobody has ever seen molecules assemble toward life. Never. It doesn't happen without a biological entity working on them. I asked all my colleagues, can you show me an example of this? Of molecules moving toward order, moving toward an ordered system where you have a non-regular assembly. Regular assembly is like A, or A, B. That you can get pure thermodynamic assembly. But non-ordered assembly is a non-regular pattern. That's what you have in DNA. We know from computer science you have to have non-regular patterns in order to have complexity for living systems. And I asked my colleagues, do you have any example of molecules without a biological entity acting upon them, moving to give an ordered assembly that is a non-regular pattern? And they sent me papers where chemists have taken molecules and assembled them in that way. I said, non-biological entity. You can't have a human doing this. Molecules don't move toward life. Well, even if you want to have molecules move toward life and you have human beings working upon them, can the human beings do it? And the answer is no. Next slide. So almost every chemical synthesis experiment in origin of life research can be summed up by a protocol analogous to this. They purchase chemicals generally in high purity from a chemical company. All right, so that's what they do. They first purchase the chemicals. They mix those chemicals together in water in high concentrations in a specific order under some set of carefully devised conditions in a modern lab. Then they can obtain a mixture of compounds that have a resemblance to one or more of the basic 4 classes of chemicals needed for life. So what you need for life is you need carbohydrates, nucleic acids, amino acids, and lipids. That's what we know. All life we know composes those 4 building blocks. So they try to make those 4 building blocks. Then they publish a paper making bold assertions about origin of life from these functionless crude mixtures of stereochemically scrambled intermediates, much like Miller did in 1952. Nothing has changed in 66 years. Nothing. Nothing's changed. Exactly where we were in 1952 is exactly where we have remained. Think about what's been done in science in the past 60 years. Think about how we have now satellite connectivity. Remember, in 1952, there had never been a satellite. Now we have satellite connectivity. We have cell phones. We have structure of DNA. We can manipulate DNA structure. Nothing has changed in origin of life studies in 66 years. That's important to realize. Then you engage with the ever gullible press to dial up the knob of unjustified extrapolations. Watch the mesmerized layperson exclaim, "You see, scientists understand how life formed." Then you encourage a generation of science textbook writers to make colorful, deceptive cartoons of raw chemicals assembling from cells which then emerge as slithering creatures from a prehistoric pond. That is exactly what is done every one of their experiments. It's all done like this. Every one of their experiments can be fit into this. And so what's going to happen is I'm going to go back and a bunch of you are going to send me articles. Look at this. These people have made life, haven't they? It fits into this. Trust me, it fits into this. Every one of them. Next slide.
So he's been through this with a lot of people complaining about different aspects, but the point is that they don't know how to make life. That's the big takeaway. And I'll send a link in the email after the fact to this talk. I think most of you would enjoy. He's very engaging and he's very direct. He doesn't beat around the bush. He says nobody knows how to do this. And he's got the chops to be credible. Here's another guy who kind of summarizes. This guy has 2 PhDs, one in teaching law and the other in political science, I think. But he just summarizes really clearly what evolution cannot do.
Evolution is based on 4 major ideas. None of them have ever been observed. Number 1, that life comes from non-life. We have not one single example of life coming from non-life. Number 2, that all the life forms that we have today came from single-cell life forms. Many people do not know that while we do have single-cell life forms that we call bacteria, we have no 2-cell life forms, we don't have 3-cell life forms, we don't have 4-cell life forms, we don't even have 5-cell life forms. That is, we go from single-cell bacteria to complex life forms. So there's no evidence that all life comes from single-cell life forms. The third postulate of evolution is that time and chance drive evolution forward, of course, and time works against evolution. The chance of one simple 100-unit protein coming together by accident this is a very big number. Time and chance make evolution absolutely impossible. The fourth postulate that evolution is based on is that what we see today, minor genetic variation in species, is evidence of macroevolution. So because cats are different colors, dogs are different sizes, that means that fish turned into cows. And of course, we know that that's not true because Mendel proved that all the genes that exist today, you can shuffle them around, but there are no new genes that create it.
So again, to summarize his 4 points, things that evolution cannot do: life from non-life, multicellular life from single-celled life forms. With all the single-celled organisms, we've got bacteria, amoebas, there's no examples of those becoming complexified. They're all the same as we see them throughout the time that we've investigated these things. We even see in some of the fossil evidence that we see very, very ancient animals and small organisms that are exactly the same as they are today. We don't see this change over time, assuming that the fossil record indicates time instead of order of burial, but that we won't go there tonight. And then life from time and chance and major species changes from small microevolutionary changes. You get big changes. I mean, you're talking about all lobsters to leopards. You just can't do that with small micro changes. Okay, so how do you build a cell? There's only 2 possible ways that a cell could come into existence from not cell. So you got not cell, oh, we have a cell. How do we get there? There's 2 possible ways. You structurally build it, and we can see from what James Tour is talking about and others that that's probably not a way to go. But there's the other possibility of chemical necessity. Does the chemistry force molecules into an order that then gives us life? And we're going to talk a little bit more about that next week. But we're talking mostly tonight about structural development, and we've seen that that's really not a good explanation for how we get cells. So this is going to be kind of our summary. This is going to be one of those simple cartoons that makes a really good point. Okay, so ready for a long story short? Okay, here we go. Oh, I need to define a term. So biopolymers, they're going to talk about biopolymers. And this is one of those terms that scientists like to use that's really big and sounds complex. And it just means bio is life, and poly means many, and molecules is an M, so biopolym doesn't sound very good. So like if you go to 121, we call you sometimes a 121er. Well, this is a biopolymer, okay? So it's just a bunch of things together to make a really big molecule. That's all it is. But they'll talk about this.
All right, imagine we're a long time ago and life isn't around yet. We've got some of the building blocks needed for life just hanging out. What are the next steps in chemical evolution that we would need to turn these little dudes into something actually living? Forming biopolymers. What's a biopolymer? That's when our building blocks team up and link together in long chains. This is what life's molecules are made out of, from DNA and RNA to proteins. Everything needs them to live, so biopolymers are kind of important. In order for chemical evolution to have taken place, nature would need a way to assemble biopolymers. But like hiding your tuna sandwich in a room full of cats, it's a very hard thing to do. Some scientists claim that it was no big deal though. They contend that under the right conditions, making biopolymers is easy enough, and from there, life could flow naturally. But is this really the case? Let's take a look more closely. These building blocks are kind of like fish. They don't have legs. And without something to float around in, they can't get around like they need to in order to do stuff. Because all known life requires water, it is commonly seen as the solution to this problem. Water allows these molecules to bump around and knock into each other, which could help link the monomers into a chain. So, water is needed, but water is actually a big problem as well for two reasons. Like bringing your mom with you on a date, water can slow down or stop chemistry. Water molecules dilute the building blocks, pushing them further apart from one another, making it less likely that they'll be able to interact. Also, water actively breaks down these polymers. Bazillions of water molecules are speeding around and constantly smacking polymer chains around. This frequently breaks the bonds, cutting the chain to pieces, something called hydrolysis. We need a liquid for these building blocks to link up, but the liquid also prevents and destroys the links. That's what makes it a paradox. Some have suggested that wet and dry cycles, or salts, may be able to fix some of these problems. However, wet-dry cycles actually degrade DNA or RNA by removing nucleobases, creating abasic sites. Formation of a biopolymer chain is actually very difficult in wet-dry conditions. Proteins in particular have an additional challenge. Like a balloon animal that relies on air to support its shape, proteins rely on water to support their complex three-dimensional form. When you remove water from a protein, it tends to fall apart, something called denaturation. Many proteins will just irreversibly collapse when dried out. Well, if water is such a problem, what if we just used a different solvent? Geochemists laugh at the idea of large amounts of a different solvent on Earth. We just don't have pools of formamide or methane or whatever lying around. Also, all of the other potential solvents tend to be very volatile as well. They evaporate quickly, or they're only produced in low concentrations, or they're only liquid at temperatures that are incredibly hostile to life, or they're nonpolar, or they're typically produced by biological activity. In other words, there are a lot of problems with using something other than water, and you can't get many of these solvents needed for life unless you already have life. More complex molecules frequently have something called chirality. You can have the same molecule just in different forms, like you have two hands, one left hand and one right hand. All but one of the 20 amino acid building blocks have chirality. Except for glycine, he's pretty silly. The crazy thing about life is that all proteins, all DNA, all RNA, all cell membranes must have the correct chirality or else life would cease to exist. Think of it like molecules having an evil twin. Here is a portion of insulin. According to this paper, if any one of these had the evil twin, the wrong chiral form, it could no longer form the 3D structure properly. If you have a long molecule like DNA, you don't just need the right chirality once. It has to be correct in every one of the 500,000 nucleotides in the simplest free-living form of life. Well, what about these papers? They show ways to preferentially filter or select one chiral form over another. So I guess it's not that big of a deal. Actually, the most successful proof of concept is from this paper, but it's an open secret in the field that the chemistry that's required for this method couldn't have happened on a prebiotic Earth.
A lot of people, our group included, are trying to find more prebiotically relevant reactions that could do what the SOI reaction does. So far, we haven't been able to find one, so that's a kind of a holy grail.
This experiment and others like it are interesting, but basically classified as prebiotically irrelevant. Natural processes are well known to produce equal portions of the two different chiral forms. That's what nature does. Filtering out only one form over another is very unnatural. There's no process that we know of on the prebiotic Earth that could have done this. But it gets worse. It's not as simple as having an evil twin, just left or right-handed chirality. Adenosine, for instance, has 15 evil twins, or 16 chiral forms. It's the identical chemical formula to the other nucleosides. It's still C10H13N5O4, but if you slot one of these evil twins into a creature, bad stuff happens. Fun fact. Platypus venom contains a protein that can flip amino acids of its victims to the wrong chirality, causing devastating pain. You dirty little chirality flipping platypus. Every biopolymer has a chirality problem. So how does modern science deal with it? Well, they largely ignore it. You could read thousands of pages in college-level textbooks or popular works, specifically on biology and the origins of life, and never even come across the concept of chirality. Diagrams that we find in our textbooks may make it seem like there's only one way for things to link up, but linking together different building blocks is hard. The reality is that there are far more ways for these little dudes to link together incorrectly than correctly. Specifically, there's 4 different kinds of ways that it can go wrong. Number 1, intramolecular cyclization. If the bit at the end links to the bit at the front, making a circle, it's lights out for that polymer. He's dead. Number 2, heterogeneous impurities. Interfering molecules or the wrong molecule could again spell death for a fledgling polymer. 3, branching malformation. You could have the right thing but have it linking to the wrong spot on another building block. Or number 4, premature truncation. There are monomers that act as terminators. Not that kind of terminator. They prevent further growth. Polymers can go wrong in so many different ways, and the longer one gets, the probability of achieving correct links goes down exponentially. Anyway, with all of the smart people working on this problem for so many years, how's the progress coming? Let's take a trek through science.
Trek through science!
Well, in the 90s, they were able to make a chain, a polymer, that was 11 monomers long. More recent experiments showed that they were able to get a chain up to 50 monomers long. Progress. Unfortunately, more recent papers have shown that these claims were wrong and not reproducible. But regardless of whether they've made a polymer that is this long or this long, even that is with extensive coddling and unnatural cheating. See this video for more information about that. When we remember that the simplest known free-living organism's genome is more than 500,000 nucleotides long, the argument sort of becomes moot. Life is so far in the distance as to be imperceptible. If, by some miracle, a polymer was able to contend with the water paradox, the chirality problem, and linkage issues, it's got a whole lot more to worry about. It has to face deadly heat, UV rays, mechanical wear and tear, ionizing radiation from the Earth, free radicals, and other forces waiting to pounce and break things down. But if you had enough time, though, surely anything could happen, right? Meet Manfred Eigen. He realized that the information in RNA or DNA that's required to maintain life would experience an error catastrophe without complex enzymes that can recognize and fix this damage to DNA. Without these error correction systems, you would essentially die of old age before you were even born. Fortunately for us, all living things are equipped with complex repair mechanisms. Unfortunately for abiogenesis, these mechanisms need to be encoded within DNA, but the DNA can't exist without the repair mechanisms. This is Eigen's paradox, a chicken and egg problem that has been around without a solution for decades. We can't have long strands of DNA or RNA unless it can repair itself, but it can't repair itself unless you have long strands of DNA. Producing any biopolymer through purely natural processes is a huge, frustrating, intractable challenge to science. But life is not based on a few random biopolymers, though. Life requires thousands and thousands of highly specific biopolymers with very precise arrangements of monomers. Even if you could get biopolymers to form by natural processes, keeping them intact is quite another challenge. RNA is so unstable that it needs to be kept in deep freeze, away from sunlight, and it has to be thrown out if it's left at room temperature for more than just a few hours. RNA spoils about as fast as a tall glass of milk on a hot summer day. And we're supposed to believe that molecules like RNA not only existed for millions of years, but also replicated and became more complex. You can believe that if you want, but it'll be in spite of the evidence, and not because of it.
So the first video that we showed with the gentleman that was debunked last week, where they go with the origin of life is to what's called RNA world. And you just saw that RNA world is not possible because it breaks down. And that's the case with many of these ideas, is that they'll have some snippet of possibility, but then when you look at the actual evidence, it can't work that way. So anyway, some things we will talk about in future sessions are the limits of what evolution can do. And we're going to talk about probably next week on this idea of pre-programmed limited adaptability, which is the idea that organisms like us have pre-programmed adaptability built in. And so certain animals have the ability to change in response to their environment, but only so far. And if the environment goes farther than they can adapt to, or they have to adapt more quickly than that organism can adapt, you're dead. And so, but that does not mean that you can have small changes leading to big changes in macroevolution. So we didn't cover a lot of things. This is a really complex field. I don't mean to oversimplify it. I don't understand it. I'm a historian, not a biopolymer maker. But I can read people and I can listen to people like James Tour and Stephen Meyer who've done the research, and I trust what they're saying because it makes good sense. So we're not going to talk about all of these things, but we'll talk about some of them in passing at levels that I can understand, which hopefully means you can understand them because you're smarter than me. So I encourage you, do some research if you're curious. If you look online, you're going to find all kinds of things. So you'll find a video that says evolutionary biologist reacts to creationists' arguments. And they're, of course, going to say that there is zero possibility of life arising by random chance. That's the creationist argument, and they're going to respond to that. On the other side, 7 scientific reasons why Darwinian evolution is a myth. And then you'll have another set of videos, Neil deGrasse Tyson and Richard Dawkins, who agree with each other on everything, so you won't find any disagreement there, on were we designed, and of course they're going to say, no, it was an accident. And you'll find Michael Behe, is there evidence against evolution? You'll find how we found out that evolution is true, from this guy that I don't know, I've never heard of. And these scientific papers destroy evolution. So you'll find all kinds of stuff on both extremes. I encourage you to look, consider the sources, be careful about which ones you choose, but you're going to find all kinds of stuff. So feel free to email me with all the debunking of everything I've said tonight, but there's probably another video out there that says the opposite, so just a word to the wise. So these conflicting claims, the question is, are they based on evidence, first of all? And secondly, are they really competing worldviews? Are they assuming that the world is only made of material things, and therefore this must be the case? Or are they open to the possibility that if the most logical explanation is something outside the box, outside the universe, a supernatural force, are they considering that as the best explanation and evaluating that possibility? In most cases, they're not, and we'll find that most of those claims are not based on evidence. They're really just different worldview issues. So, just wrapping up, we talked about how the materialists explain the world and looked at a little bit more of a workable idea, hopefully, We looked at the introduction to intelligent design. We'll go a little bit more into intelligent design next week, and we did the long story short. So next week, we'll do a little bit more, and you can see this was my original outline. I show you this to show you that this is morphing. So we covered some of intelligent design this week. We're going to cover more of it next week and cover more examples of intelligent design. So I think that'll be kind of a fun week, a little lighter weight than this week was. And then we'll talk about biological wonders. We're going to talk about things as diverse as honeybees and hummingbirds and whales, to just show how some of these unique design features could not have been mutated from other organisms, because they don't exist anywhere else. And they're just ingenious designs to survival. And then we'll work our way toward the solar system as a design example. And then we'll talk about the idea in last week of a whole new argument that's just come out in the last couple of years on systems engineering, how you build a system of systems that are all dependent on each other. It's the ultimate chicken and egg problem, and I think you'll enjoy that. So let me open it up for questions, and we'll wrap up. Anybody have any questions that we should cover this week? Or did I already answer all of your questions? Are you overwhelmed? I'm overwhelmed. Okay. Well, we'll wrap it up here. If you have questions, I will be here. I want to thank Jason, who's been helping in the sound. He and I have been tag teaming these design and the flood series. He was the lead on the flood series. I've been the lead on this series. But we've been a great duo, and I really appreciate Jason's help. So thank you, Jason. Let me pray for us, and we'll dismiss. Lord, we thank you that you are a God who told us what you did, and that we can believe that because there's evidence showing that that's the case. Thank you for men and women who have done the design research to validate that the Bible is actually the best explanation we have for life and for the universe and for all the forces within it. Thank you that the heavens declare the glory of God, and that we can declare that along with the things that we see around us, and we just proclaim in awe that you are the God of the universe. We thank you for our time tonight in Jesus' name. Amen. Okay. Thank you.